Let's examine the anatomy of some of the external structures of the male reproductive system on our specimen. The testes are suspended outside the body wall within a pouch of skin and connective tissue called the scrotum. The scrotal sac has been opened on the left side to expose a testis, whereas the right side of the scrotal sac is still intact. The scrotum consists of several layers of tissues that are derived from the anterior abdominal wall when the testes migrate from their fetal location inside the body to their location outside the abdomen. It is important that the testes undergo this migration because viable sperm will not be produced at internal body temperatures and will develop only at cooler temperatures found within the scrotal sac. Two muscles associated with the scrotum help to regulate the temperature of the testes. The cremaster muscle is formed from skeletal muscle fibers derived from the internal oblique muscle of the abdomen. It reacts to decreasing temperatures by contracting to elevate the testes toward the trunk. The dartos muscle, consisting of smooth muscle fibers, wrinkles the skin of the scrotum in response to cooler temperatures. This action reduces heat loss around the testes. To reveal a testis, the left side of the scrotal sac has been opened. Each testis is about 2.5 centimeters in diameter and 4 centimeters in length. The layers surrounding the testis are derived from structures associated with the abdominal wall. One of these coverings is the tunica vaginalis, which is derived from the peritoneum. Deep to the tunica vaginalis is the tunica albuginea, the dense fibrous capsule of the testis. Inward extensions of the tunica albuginea delineate 200 to 300 individual lobules. Each lobule contains 1 to 4 tightly coiled seminiferous tubules, which are the sites of sperm production. 3 cell types are associated with the seminiferous tubules. In this illustration of a cross-section of a seminiferous tubule, we can identify these 3 cell types. Lying in the interstitium of connective tissue between adjacent tubules are the interstitial endocrinocytes, or cells of Leydig. Their most important secretion is the hormone testosterone. This hormone plays several different roles in the development and function of male reproductive organs throughout puberty and adulthood. Another cell type that is found inside the seminiferous tubule is the sustentacular, or Sertoli cell. This cell nourishes and supports developing sperm within the tubule. The remaining cells located in the testis are developing sperm, or gametes, which are in various stages of spermatogenesis. Spermatogonia, the stem cells that divide to produce the primary spermatocytes that will undergo meiosis, lie close to the basement membrane of the tubule. As gamete development occurs, primary spermatocytes appear in the middle region of the tubule, and then the more mature secondary spermatocytes are formed somewhat closer to the lumen. The spermatids are located near the lumen, with the immature spermatozoa lining the lumen and ready to move out of the testis. Follicle-stimulating hormone and testosterone are important hormones that regulate spermatogenesis. Each sperm contains 3 regions: a head region, midpiece, and tail. The head is 3 to 5 microns long and contains a nucleus with DNA and an acrosome, which caps the head. The acrosome contains hydrolytic enzymes necessary for penetration of the egg by sperm. The midpiece is 5 to 7 microns long and loaded with mitochondria. These powerhouses produce ATP necessary for movement of the tail, or flagellum. The flagellum is about 45 microns in length. Following the release of immature sperm into the lumen of the seminiferous tubule, the sperm travel through a series of ducts in the testis to reach the epididymis. First they travel through the straight tubule, then the tortuous reedy testis, and finally the efferent ductules to reach the epididymis. final maturation of sperm occurs as they travel through the coiled duct within the epididymis. During this process, the sperm learn to swim and finally become capable of fertilizing an egg. The maturation process can take 2 to 3 weeks. The epididymis has 3 portions that will be identified on our specimen. The head is the region where the efferent ductules from the testis join the ductus epididymis. The body contains a highly coiled tube which continues into the tail portion. Sperm travel from the tail of the epididymis to the ductus deferens, which is sometimes referred to as the vas deferens. It is approximately 45 centimeters long and travels in the scrotal sac near the epididymis towards the inguinal canal. The ductus deferens is the structure that is ligated during a vasectomy. The spermatic cord contains structures traveling to or from the testis. These structures within the cord are enclosed in layers of tissue that are derived from the anterior abdominal wall. The spermatic cord travels through an oblique passageway in the inferior border of the abdominal wall called the inguinal canal. The inguinal canal, which has been opened in this specimen, is about 5 centimeters in length. Its floor is formed in part by the inguinal ligament. The abdominal musculature also contributes to the formation of the canal. The left spermatic cord has been opened to reveal one of the most important structures within the spermatic cord, the ductus deferens. Another visible structure within the cord is the pampiniform plexus of veins which surrounds the testicular artery. The relationship of the veins to the artery functions to cool the arterial blood as it flows towards the testes. Other structures found within the cord include nerves and lymphatic vessels. For orientation, we are looking down into the pelvic cavity from a superior view. We will identify the aorta, the inferior vena cava, the common iliac arteries, and the posterior side of the bladder. We can trace the pathway of sperm and the location of 3 accessory sex glands, which secrete fluid that mixes with the sperm to form semen. These fluid secretions provide a transport medium and nutrients for the sperm. The paired ductus deferens carrying sperm from the testes enter the abdomen via the inguinal canals and travel on each side toward the posterior bladder. As they approach the bladder, each expands to form the ampulla portion of the ductus deferens. The first pair of accessory glands can be seen in this specimen. The seminal vesicles are located behind the urinary bladder near its base. They are about 5 centimeters in length and produce an alkaline secretion that is rich in fructose, ascorbic acid, and a coagulating agent. Secretions from the seminal vesicles produce about 60% of the volume of semen. In this illustration of the posterior aspect of the bladder, we will first identify the ampulla of each ductus deferens. The union of the ampullas and the ducts of the seminal vesicles form the ejaculatory ducts. 2 ejaculatory ducts, one from each side, open into the prostatic portion of the urethra. The single prostate gland forms a donut-shaped ring around the prostatic urethra. Prostate secretions are milky, slightly acidic, and contain proteolytic enzymes which include a liquefying agent. Secretions of the prostate account for about 30% of the total volume of semen. Another pair of accessory glands, the bulbourethral glands, are quite small and are located inferior to the prostate gland within the urogenital diaphragm. The ducts of the bulbourethral glands open into the spongy urethra in the penis. These glands secrete an alkaline fluid prior to ejaculation that protects sperm by lubricating the lining of the urethra and neutralizing its acidic environment. In this illustration, we can identify the tissues that form the penis. Most of the penis is erectile tissue. First, we'll identify the urethra, which is surrounded by the corpus spongiosum. The corpus spongiosum attaches to the urogenital diaphragm at the bulb of the penis and distally enlarges to become the glans penis. The opening of the urethra in the glans penis is the external urethral orifice. A cuff of loose skin, called the prepuce or foreskin, surrounds the tip of the glans penis. There are also paired dorsal erectile tissues called the corpora cavernosa, or singularly called a corpus cavernosum. The proximal end of each corpus cavernosum is the crus of the penis, which attaches to the ischial and inferior pubic rami. These two crura and the bulb form the root of the penis. The shaft or body extends from the root to the tip of the penis. To get a better understanding of the anatomical relationships of the erectile tissue, we will view a cross-section of the penis. The urethra is surrounded by the corpus spongiosum. The paired corpora cavernosa are situated dorsally. Notice that the corpora cavernosa make up the bulk of the penis. The erectile tissue is highly vascularized and becomes engorged with blood during sexual excitement. On a hemisected specimen, we will review some of the structures of the male reproductive system that we have just described and identify the three portions of the male urethra. The bladder in the male is located posterior to the pubic symphysis and anterior to the rectum. Lying inferior to the bladder is the prostate gland. The male urethra is approximately 20 centimeters in length and functions to carry both urine and semen. The male urethra extends from the bladder through several structures to reach the exterior and contains three named portions. The proximal prostatic urethra courses through the prostate gland. The middle portion of the urethra passes through the urogenital diaphragm and is called the membranous urethra. The distal portion extends through the length of the penis as the spongy or penile urethra. The erectile tissues that can be identified in this dissection are the corpus spongiosum and the corpus cavernosum. The bulb of the penis can also be seen in this dissection. The female reproductive system is more complex than the male's because in addition to producing gametes, it is also designed to protect and nourish a growing embryo. Female gametes, called ova or eggs, are produced in the ovaries. Like the testes, the ovaries also produce sex hormones, but in this case the female sex hormones estrogen and progesterone. On a monthly cycle, one of the ovaries will release an egg. The egg is transported through the uterine tubes to the uterus. Let's examine some of the structures of the female reproductive system within the pelvic cavity. For orientation, we are looking down into the pelvic cavity from a superior view. The broad ligament, a peritoneal covering, drapes over the uterus, ovaries, and uterine tubes. There are two depressions which lie between viscera in the pelvic cavity. The shallow vesicouterine pouch is located between the bladder and the uterus. The deeper rectouterine pouch is found between the rectum and the uterus. In order to see the components of the female reproductive system more clearly, the peritoneal covering has been removed. The ovaries are located on either side of the uterus. The ovarian ligament, a specialization of the broad ligament, attaches the ovary to the uterus. Cuboidal epithelial cells, called germinal epithelium, form the outermost layer of the ovary. This layer does not actually produce ova, as you might think by its name. Deep to the layer of epithelium is the tunica albuginea. Like the external surface of the testis, it contains dense, irregular connective tissue. The outer portion of the ovary is the cortex. It surrounds the inner medulla, which contains blood vessels and nerves. The process of oogenesis, or gamete formation, occurs in the cortex. However, unlike spermatogenesis in the male, oogenesis begins before birth so that a female is born with a lifetime supply of primary oocytes, the cells that will undergo meiosis to form ova. A single layer of cells encases each primary oocyte, and the composite structure is called a primordial follicle. After puberty, a few primordial follicles are activated each month. As additional layers of cells begin to appear around the oocyte, the primordial follicle becomes a primary follicle. These cells forming the follicle are called granulosa cells. In addition, a connective tissue layer called the theca folliculi forms around the follicle. The thecal and granulosa cells cooperate to synthesize estrogens. A secondary follicle is distinguished by the presence of a fluid-filled cavity called the antrum. The antrum continues to expand, and the primary oocyte completes meiosis I to form the secondary oocyte. At this point, the structure is called a vesicular or Graafian follicle. Notice how the vesicular follicle bulges from the side of the ovary. Ovulation occurs when the vesicular follicle ruptures, expelling the secondary oocyte from the surface of the ovary. Although the ovary contains several follicles at various stages of development, through some unknown mechanism, only one mature follicle responds to a surge in luteinizing hormone to release its egg during ovulation. The ruptured follicle, the corpus hemorrhagicum, changes to become the corpus luteum, which produces progesterone and estrogens. If pregnancy does occur, the corpus luteum continues to produce hormones for about 3 months until the placenta develops and takes over this function. If fertilization of the egg does not occur, the corpus luteum degenerates and its hormone production ends in about 10 days. The corpus albicans represents the scarred remains of the corpus luteum. Hormones produced in the anterior pituitary gland influence the normal activities of the structures within the ovary. Growth and development of the follicles and estrogen secretion are controlled by follicle-stimulating hormone and luteinizing hormone. A surge of luteinizing hormone stimulates ovulation. Luteinizing hormone also transforms the ruptured follicle into the corpus luteum and stimulates its production of progesterone and estrogen. There are two uterine tubes, each about 10 centimeters long. The uterine tubes, also known as the oviducts, carry the ovulated oocyte from the ovary to the uterus. Fertilization usually occurs in the uterine tube. At one end, the uterine tube bears a funnel-shaped structure called the infundibulum. Extending from the infundibulum are ciliated finger-like projections called fimbriae. The entire structure hovers over the ovary to sweep up the ovulated egg. There is no direct contact between the infundibulum and the ovary. At the other end of the uterine tube is the isthmus, which is a short, narrow, thick-walled structure that connects the uterine tube to the uterus. The middle portion of the uterine tube is called the ampulla. The entire uterine tube is covered by peritoneum called mesosalpinx, a portion of which can be seen in this specimen. Each uterine tube is lined with ciliated columnar epithelium, and its middle layer is composed of smooth muscle. Peristaltic contractions of the smooth muscle and the sweeping movements of the cilia guide the oocyte through the uterine tube towards the uterus. The uterus is a large muscular organ which houses the developing embryo during pregnancy. The round ligaments extend from the uterus toward the anterior body wall. They continue through the inguinal canals to attach to the labia majora. These ligaments help to maintain the uterus in a tilted forward position. This hemisected specimen provides a different view of some of the structures that we just discussed. The uterus normally sits in a forward or anteverted position over the bladder, which is protected by the pubic symphysis. The vesicouterine pouch is located between the bladder and the uterus. The rectouterine pouch is found between the rectum and the uterus. Structures that we can identify include the round ligament, uterine tube, and ovary. The rounded region of the uterus above the opening of the uterine tubes is the fundus. The largest portion of the uterus is its body. The cavity within the uterus that is surrounded by its body is the uterine cavity. The uterus narrows and its cervix projects into the vagina. The external os is the opening of the cervical canal into the vagina. The outer covering of the uterus is the layer of visceral peritoneum called the perimetrium. The myometrium, a thick layer of smooth muscle important during childbirth, forms most of the substance of the uterus. The innermost layer, the endometrium, lines the uterine cavity. Contained within this layer are numerous glands and blood vessels which prepare the uterus for implantation of the embryo should fertilization occur. The endometrium is divided into two different regions or layers. The stratum functionalis is the layer of endometrium which is shed during menstruation. The deeper layer, the stratum basalis, is not shed, but instead produces a new functional layer after menstruation. The arterial supply to the uterus is rich with blood being supplied from both the uterine arteries and, to a lesser extent, the ovarian arteries. As the uterine artery enters the uterus, it branches into arcuate arteries within the myometrium. These arteries give off radial arteries. Radial arteries furnish straight arteries to the stratum basalis and spiral arteries to the more distal stratum functionalis. Changes within the spiral arteries in the functional layer are responsible for the loss of this layer during menstruation. In this hemisected specimen, we can identify the vagina, which is a muscular tube about 10 centimeters in length. The vaginal canal serves as a passageway for menstruation and childbirth and also receives semen during intercourse. At its superior end, the vagina surrounds the cervix of the uterus. Because the diameter of the vagina is larger than the diameter of the cervix, recesses are formed around the cervix. Each recess is called a fornix. Vulva, or pudendum, is the term used to describe the female's external genitalia. The mons pubis is a mound of adipose tissue overlying the pubic symphysis. Posterior to the mons pubis is a pair of folds consisting mostly of adipose tissue called the labia majora. The labia majora are the homologs of the scrotum in the male. The mons pubis and the external sides of the labia majora are covered with hair after puberty. Located medially to the labia majora are thin folds of hairless skin called the labia minora. The area enclosed by the labia minora is the vestibule. This region contains the external urethral orifice, the opening of the urethra, and the vaginal orifice, the external opening of the vagina. The clitoris is the homolog of the male's penis. The skin of the labia minora forms a hood over the erectile tissue of the clitoris, the prepuce, which is homologous to the foreskin that covers the distal end.